putting together this very interesting workshop, and the introduction from Mikio today actually kicked in me an idea for a workshop next year, the topic of which will be how can we control latent neural states of voter to influence outcome of elections. I'm not sure if it's a good idea or not, but let's stick with the idea of this year's workshop and think about how do we interpret these internal neural states which we learn these models from data. So let me back up and kind of take a step back and think about how we derive theories in computational neuroscience, or how did we do it traditionally. So if we had recordings from animals, traditionally we would start with looking at average firing rate. And this is an example from an LIP neuron recorded from a monkey during a decision-making task. An LIP neuron shows a beautiful ramping activity. You see these traces on the right, rising from low activity to high activity over time, and different traces show different stimulus difficulty. And interestingly enough, these activity traces resemble very much the average activity traces of a behavioral model called the drift diffusion model, which was introduced very nicely yesterday by Scott Linderman. And the idea is that subjects' behavior can be described by a process which is noisy, so there is some noisy diffusion process going on, but there is also a drift or bias towards one of the decision thresholds, a bounds, such that when you hit one of these threshold, decision is committed. And usually, by this resemblance of trial average neural responses to behavioral models, we would conclude that this neuron is involved in performing this computation. It's performing in realizing evidence accumulation in the brain. However, it has been realized that if we start to look in many neurons, even if we keep looking in trial average responses, and this is a data set from M. Churchland's lab where they recorded neural activity in rats during decision making, and these are just 3 example neurons. What you realize, their profiles don't even look like the ramping activity. Each and every one has a very idiosyncratic time course, and it is really hard to reconcile this heterogeneity of neural responses with the idea that each of them is performing the same behaviorally relevant computation. And in addition, it was also pointed out that when we look in trial average responses, maybe we're missing something. And these drift diffusion traces, which we would hypothesize are relevant for describing spiking activity of single neurons, can be only one explanation. You can also construct a model, which was beautifully pointed out by Kenneth Latimer and Jonathan Pillow, which would have very different dynamics on single trials, but the same trial average responses. And just by looking at spike trains, it's not very obvious which of those models is a better model for the data. In such situations, we also had a very classical approach how to deal with those models. And this paper, which I just cited, beautifully illustrates this approach. We have 2 a priori hypotheses about how this brain area, how this circuit could function. For example, one hypothesis could be that neurons perform evidence accumulation and their responses are consistent with drift diffusion model. Or an alternative hypothesis could be that this model shows discontinuous firing rate traces on single trials, which is consistent with a step-like or jumping model. Then we can feed both of these models to the data before model selection and decide which of these hypotheses is more consistent with the data. After this paper had been published, it was a whole line of follow-up research arguing about shortcomings of such model selection. But even without going into technical details of model selection, you can realize a very fundamental problem with this approach. That we start with a priori hypothesis, and there is no guarantee that correct hypothesis about what this brain area is doing, it cannot be even among the set of hypotheses which we consider. And therefore, recently, a new idea started to appear in the literature. Instead of starting with very rigid but very interpretable, simple models, we can construct a very flexible model. And by flexible model, I mean that in a single model architecture, this model can realize very different, a broad class of hypotheses. An example of such a model is a neural network, for example. In a recurrent neural network, by changing parameters of this model, we can realize very different kinds of dynamics. So the idea now is the following. We will feed this flexible model to the data, and then we will analyze the model to extract hypotheses directly from the data. So it's a purely approach to hypothesis discovery or to theory building bottom up from data. However, here is still the question, how do we optimize these flexible models? And usually what we do, as has been mentioned in entire row of previous talks, we optimize these models for predicting new data, right? So we do cross-validation, we use data which was not used for fitting the model, and the idea is if the model can predict the data very well, it should be the right model. However, we never ask the question to what extent these flexible models optimized for predicting data provide the accurate and correct interpretation of this data. In other words, whether hypotheses which we derive with this approach do actually match the biological ground truth. And this question was difficult to answer with flexible models, such as recurrent neural networks, for example, because they lack this internal interpretation. And what I mean by that the correspondence between a parameter set of neural network and a hypothesis which this parameter set represents is not immediately obvious. And usually we have to go through very complex steps of multiple additional calculations to interpret this model. And therefore comparison with ground truth was challenging. However, the history of science teaches us that good data prediction and correct interpretation do not always go hand in hand. And here is an example from years ago of two models of a solar system, one by Ptolemy and one by Copernicus. And one model placed the Earth in the center of the solar system, and another one placed the Sun in the center of the solar system. And because these models were very fine-tuned with kind of very tiny additional details, both of them made very accurate predictions of the movement of celestial bodies. However, it is very obvious that interpretation of these models is very different. So what I will show you today is a flexible but intrinsically interpretable modeling framework for modeling neuropopulation dynamics, which we developed. And because this framework is intrinsically interpretable, it allowed us to perform a direct comparison of models which we learned from the data with the ground truth on synthetic data from which this data was generated. And what we discovered during this procedure is that there is a disconnect between the generalization performance of flexible models and their interpretation. That not all models which generalize well will give you the accurate interpretation of the data. And therefore we had to go and think beyond this idea of generalization and develop an alternative approach for discovering models with correct interpretation, which relies on consistency of features which we discovered from different data samples. I guess this idea was introduced already multiple times in this workshop, that instead of looking in single neurons, we are looking in populations, and changes of activity of one neuron correspond to coordinated changes across the population, as shown by the multiple axes on the right-hand side. And usually we deal with situations where this population dynamics is low-dimensional, which means we only need few axes to capture a lot of variability in the data. And the question is, how do we now reconstruct dynamics in this latent state from the data? So the approach which we developed is a nonparametric. So here is an idea of how it works. So imagine you have very complicated trajectory in this latent space. How can we understand it? Only if we knew the governing equations of motions which generated these trajectories, we could interpret the system immediately, because we have the entire dynamical system theory, dynamical system tools to understand models written in this form. So for example, you can think about right-hand side of this equation as an evolution in some energy landscape, in some potential landscape, where valley represent attractor states and trajectory has easier time rolling down the valleys and climbing up the hills. But in multiple dimension, in addition to this potential landscape, there is also additional component which is called curl flux, which can result in non-stationary motion between these attractors. So now imagine that f of x, the right-hand side of this equation, can be represented by any arbitrary continuous function, and we can morph the shape of this function in any way, searching through the space of all possible continuous functions which can be placed in the right-hand side of this equation. So now what we will do, we will search or fit through the space of continuous functions to discover the model which best describes the data. So here is a schematic of framework. So we make an assumption that population dynamics can be described as a dynamical system in the low-dimensional latent space, and each point in this latent space corresponds to particular firing rate of every neuron in our population. And these functions, f of x, I call them firing rate functions. You can think about them as a tuning function of every neuron to the position in the latent space. It's very much like what Anki presented in her talk. So the idea is the responses of individual neurons can be very heterogeneous because of heterogeneity of these tuning functions, and nevertheless, all these neurons have a common population dynamics. To account for variability of spiking, we also add a Poisson component, so given instantaneous firing rate of each neuron, spikes are generated as Poisson processes. What we will learn from data, we will use gradient descent procedure over the space of continuous functions, which represents the tuning curves as well as driving forces in the latent dynamical system. Let me illustrate on synthetic data how this approach works, but first I will make comparison with other existing methods which rely on ad hoc assumptions about latent dynamics. This is a synthetic data where I have 16 neurons over time, and the data was generated from a very simple scenario where firing rate or population state of this neuron alternates between two states stochastically over time. And similar kind of dynamics we observed previously, for example, in visual cortex of monkeys. If we would try to infer these dynamics with other methods, and for comparison, I show here two methods which make very opposite ad hoc assumptions about what these dynamics can be. One of the methods is a Gaussian process factor analysis method, which also was mentioned in multiple talks. And Gaussian processes make explicit assumption that latent dynamics are smooth. And although this data was generated from very discontinuous transitions between two well-defined states, the Gaussian process analysis will reveal us smooth fluctuations rather than discontinuous transition. On the other hand, if I use a hidden Markov model, which makes just the right assumption that there are two latent states and discrete transitions between them, we would make very accurate inference. However, it requires an ad hoc guess on our end to get the right dynamics, but any post hoc analysis can actually not tell the difference between two models, and by any post hoc analysis, you would not be able to distinguish which of them is better describes the data. On the other hand, we can be in a situation where the latent process is more smooth and closer to the Gaussian process assumptions. And if we fit hidden Markov model in this situation, we would still discover discrete states, and by post hoc analysis, we would be very convinced that this is the right answer. So in contrast, when we use our flexible framework and fit it to data which was generated from discrete transitions, we discover these discrete transitions, but with the very same model applied to a data which evolves more continuously, we are not fooled and we discover smooth trajectories. So how does our framework achieve it? So here's the idea. As I mentioned to you that we fit the shape of the potential landscape, and in this case, dynamic is simple and one-dimensional. The switching dynamics corresponds to a potential landscape which has two discrete wells, two discrete attractors, and noise drives the system between two discrete attractors to generate these discontinuous transitions. The smooth trajectory, on the other hand, corresponds to fluctuations around a single attractor well, resulting in these more continuous traces. I will show you now a brief movie of our gradient descent optimization algorithm, which starts with an unspecific guess, and the current model estimate of the potential landscape shape will be shown with the red line, and the gray line shows the ground truth from which we generated the data. And as you can see through the iterations, the shape of this landscape smoothly morphs until it arrives at the right solution from the data to which it was fed. And as I mentioned to you, simultaneously with learning this potential for the latent dynamics, we can also learn firing rate functions for the neurons which are in our population. I will not focus in the sake of time on firing rate function inference. I just show one slide illustrating that if we have a population of multiple neurons with nonlinear tuning functions, we can infer this from data simultaneously with dynamics, and we have a good correspondence between learned tuning functions and the data and the ground truth from which data was generated. So far, so good. I introduced you the framework and I showed you that it works. However, there is a caveat. When I showed you the gradient optimization movie, we stopped at some particular iteration. It was iteration 530, but there was no reason to stop at that particular iteration because actually gradients do not disappear at that point. So I can keep optimizing my model, and let's keep watching this movie and see what happens if we run it for a couple more iterations. You see how models start to deteriorate and develop features which were not present in the ground truth potential. It develops additional wells, and it looks nothing like the ground truth. So this is maybe not surprising, although it was surprising to us. Initially, we were more optimistic venturing into this enterprise, but it is a phenomenon which is known as overfitting. What we have here, we have a finite sample of very stochastic latent trajectory, which we observe only through a finite sample of Poisson sampled spikes. So it's a lot of noise in this data, and the model needs to optimize likelihood of this data, and apparently developing these like tiny wiggles in the potential leads to higher likelihood of this particular realization of spiking noise, and that's why model comes up with this idea. Here is just another illustration of the same thing. So this is the likelihood of iterations of the gradient descent, and these are iterations on the x-axis. And on the right-hand side, you see the shape of the potential along this path of optimization. And although we come very close to the ground truth potential somewhere in between, later on we start to deteriorate and develop spurious features. So our response to overfitting, or our response to model selection in this case, is usually cross-validation. So what we should do, we should generate another sample of spikes, which was not seen by the model during optimization procedure, and by evaluating likelihood on this new data, we will be able what the right model should be. This is exactly what we did. So what you can see here are two traces of log likelihood. One is on the training data set, and another is on the validated data set. And one striking feature which you can realize from these traces is that there is a very long plateau in those two likelihood curves, right? So you don't see a very prominent minimum in the validation set. And remember that any of these points along this optimization path is a different model, which has very different features of the potential, meaning it has very different interpretation. But nevertheless, all those models are able to predict new data nearly equally well. And the reason for that is that these new spurious features develop on top of the correct features, which are discovered first, and they do not harm generalization performance of those models. Curiously enough, very similar behavior was reported also for neural networks, that they they tend to develop these spurious features on top of correct features, which are learned first. So let's try now to pick the model which will give us the best validation performance. Let's pick the minimum of this validation curve. And the minimum in this particular simulation would be somewhere around here. And what we will get a very overfitted model. And the reason for that is that our validation set, just like our training set, is a finite sample of spikes which contains a lot of noise. And we are dealing with selection of models which are all nearly as good in generating very similar statistics of spiking. And therefore, just by chance, one of these models on a finite and noisy validation sample can happen to be the best. So if we repeat this exercise on multiple realizations of training and validation data, on some occasions, we recover the ground truth models using the cross-validation performance. But in other cases, we get models which are very well overfitted. This behavior disappears if you have enormous amounts of data. So first of all, if you have a lot of data, and we did these simulations, let's say, for example, resampling Poisson spike train on each iteration of gradient descent, you will not overfit in the first place. But the more data you have, of course, the overfitting becomes less and less likely also at the stage of model selection. However, with realistic amounts of data which we have from neurophysiology experiments, this behavior is very common. And although these models can predict new data very well, we cannot reliably interpret these models because their features differ from the ground truth features from which they were generated. So at this point, we had to think about how now can we select the model, not for the ability to predict data, but for the ability to provide correct interpretation of the data. And we realized that we have to give up this idea of generalization altogether. So the idea which we had is actually relatively simple. we can still divide our data into halves, which are usually used for training and validation of the model. But we can use these two halves differently. So the reason that the true features of the data, which we will discover in two different halves of the data, should be identical. But the noise features, which result from overfitting to noise pattern, are very unique. So the overfitting pattern is very unique to a particular noise sample in each piece of the data. So if we compare features we discover in two halves of the data, then the true features should coincide, while noise features will diverge. And this is exactly the idea which we had for identifying models with correct interpretation. However, the difficulty is that on different data samples, when we run optimizations, the same features will be discovered at different iterations. And here is an example. This is a simulation where I have 10 different samples of data, which are all generated from the same ground truth models. And this is more or less the same potential shapes on the right-hand side. And they are found at this iteration showed by dots on the left-hand side. Right? So we cannot, let's say, take all models which are discovered after 100 iterations and compare them to each other, because it would be meaningless. We would be compared underfitted potential to completely overfitted potential. So we had to have a procedure which would allow us to align those potentials and compare apples to apples. And for this, we developed a method of measuring feature complexity, the complexity of features which are learned at each state of gradient descent optimization. So what we call feature complexity, or we choose this measure, it actually comes from physics. It was not developed by us. So we use a measure which is an entropy of trajectories generated in a potential of a particular shape. So the idea is the more structure the potential has, the more features will trajectories in that potential have, and the result of that, we will have higher feature complexity measure. So here I plot feature complexity now for a particular simulation as a function of the gradient descent optimization. And what you see, we start with very simple potentials, and over the course of optimization, we develop more and more features, and this measure grows over the course of optimization. And the dashed line here shows the ground truth feature complexity. And you see that first we are learning the true features of the model, but at some point we cross this line and start to overfit to noise, and we start to develop spurious features. So in real data, we don't know where this dashed line is. We don't know the ground truth. And the difficulty is that we take different data samples, they will cross this ground truth feature complexity on different iterations of the gradient descent. However, what we can do, we can align those models along the feature complexity axis. So what this plot shows, it shows a generalization log likelihood or validated likelihood as a function of feature complexities for these two different simulations. And you see that these curves perfectly align to each other, because we align potential by plotting this as a function of feature complexity. We basically align the potential shapes across the simulations. So what happens now if we pick a potential somewhere here, which has relatively low feature complexity? We will see that across these two data samples, the discovered models will tightly overlap, because the simple features are the same between two data samples. If, on the other hand, we pick a point here along this axis which has high feature complexity, we will find potentials which look different between two simulations. And interestingly enough, if we traverse this axis and we track the consistency of features until the point when they start to diverge, the point when these potentials start to disagree with each other, coincide exactly with the ground truth feature complexity. Yes?
How sensitive is this to initialization? Like if you start with a very complex well, does it align correctly?
So like we do here a little trick, because we also knew about feature complexity measure from the outset, so we always initialize our procedure. This feature complexity measure, it's nice because it's convex, it has a unique minimum, and we always initialize our optimization with a model of least complexity. All right, so now let me illustrate that this procedure works. So this is another kind of reiteration that best validation is not the right way to go. These are 10 simulations. These are validated log likelihoods. Dots show the minimum of validated log likelihood, and this is a corresponding potential for the ground truth, which was a double well potential. And you see we can discover very different models using this procedure. But on the other hand, the feature consistency allows us reliably identified models which agree with ground truth. And if we go beyond, we again start to have potentials which disagree. And in the final couple minutes, I would like to illustrate that this phenomenology of overfitting is not only kind of specific to our synthetic data, it is also the same phenomena we also observe on the real data. An example I will give here is recordings of spiking activity from the visual cortex of monkeys, which were performed by Nick Steinmetz and Thierry Moore's lab a couple of years ago. And Nick used laminar recordings to record across depths of single cortical columns. And the phenomenon which we kind of noticed recently and analyzed recently is that activity is very synchronized through the depths of cortical columns and alternates continuously between periods high and low firing rate, which are nearly synchronous across all the channels. This is what we call tonal dynamics. And previously, we analyzed this data using hidden Markov models. However, the hidden Markov model, although it was useful to learn a lot about those dynamics, it didn't allow us to answer the questions: are these fluctuations more consistent with really metastable transitions between discrete attractor states, or are these just some fluctuation around a single attractor state, which several existing models of neural variability would suggest? So our approach is very good to answer this question, but also because we didn't have ground truth on this data, what we decided to do, we decided to fit activity of each channel separately, and if we would discover the similar shape of potential across every channel, it would give us some confidence that the model which we learn has something to do with their population dynamics. So first of all, let me show the validation curves. So we divided data in two halves, D1 and D2, and we also fit the entire data set. And this is a validation curve for one particular recording, and you see the same feature which we saw on synthetic data. Very flat generalization plateaus, which, by the way, if you ever fitted large neural networks exhibit the same kind of long generalization plateaus. Now, if we pick minima of these validation curves, we get potentials which disagree between different partitions of the data. On the other hand, we used our procedure of taking feature complexity, consistency, and a little bit by design of our method, we discover potentials which tightly agree between different data parts. But also, it was interesting to see that what we discover is a bistable potential, suggesting that these transitions, which we previously analyzed in the visual cortex, are consistent with metastable dynamics. And when we repeated this analysis across all 16 channels, although there was some variability across channels, overall, I would say there was a good consistency. All right, so just to recap, I showed you today a flexible and at the same time intrinsically interpretable framework for modeling neural population dynamics. With this framework, we found that there is a disconnect between generalization and interpretation of flexible models, and we proposed a procedure for inferring models with correct interpretation. I would like to thank everyone in my lab, and in particular Mikhail Genkin, who was spearheading this project and is really the lead on this project, as well as my funding sources. Thank you.
Hi, if you could go back to the on-off results and looking at the validation versus feature complexity plot. Yeah, that one on the top right. I was wondering how you chose that point on the curve to be where you stopped versus any other part on that curve?
Right, so initially we used to choose this point by eye, but reviewers didn't like it. So we had to come up with a procedure to develop a more principled way. So now we develop a procedure which is using Wasserstein distance between two probability distributions. So each potential corresponds to a probability, equilibrium probability distribution in that potential. So we just measure the distance between this potential, and by placing a threshold on this measure, which turns out to be relative arbitrary because it's like the consistency is very high early on and then it starts to grow. So like it's not like very critical where you place this threshold, because qualitatively you will still get the same model, so you will only get maybe slightly more complex or slightly less complex double bound potential. Yes?
So have you tried using any regularization techniques to reduce overfitting?
Yes, we did, and you get the same issue. I even have a slide for this if you're interested. I don't know how to pull it up very quickly, but the idea is, okay, let me pull up the slide. Maybe just in old-fashioned way. Oh, here was the regularization slide. Right, so you can also, we can use the same, let's say, metric, let's say trajectory entropy measure as a regularizer across the models. However, now we have an additional hyperparameter, which is the regularization strength. we need to decide how much do we weight data likelihood and how much do we want to penalize model for to be complex. And usually hyperparameters are chosen by cross-validation, and this is what we did, and these are the same two data samples which I showed you before. And as you see, in one case, we pick an overfitted model, and in other case, by choosing the strength of regularization using cross-validation, we will pick the right model. But the behavior is so early stopping is also a regularization technique in a way. So it doesn't matter which regularization technique you use, if you have finite data, you will have this problem. Yes?
This is a very nice approach, but so far you've had 1D low dimensional domains. So do you have any idea how to scale even just to 3, 4, 5? Especially because what you haven't talked about here, right, is the fitting of the single neuron tuning curves, which have to happen together with this.
Yeah, I have a backup slide for this as well. Yes, so we can scale it up. We can scale it up to 2 dimensions, maybe possibly to 3D, but I don't even want to think about. 2D is very straightforward, right? So the thing is that we have to solve a partial differential equation during optimization procedure, which doesn't scale very well to high dimensions. So I think we will never literally go a bit beyond 2D. However, the question about how many dimensions do I need to capture neural data is very much related to Anki's talk yesterday, right? And this is a question about nonlinear dynamics versus nonlinear embeddings. Imagine you have a very simple situation, something like place cells, and you have a one-dimensional trajectory and a population of place cells which code this trajectory with these Gaussian-like peak place fields. So what would happen if you do not allow your tuning curves to be nonlinear? It would happen that you require multiple dimensions to capture this data, because if you go along a single dimension and your firing rate starts to increase, you cannot bring it down along the same dimension if you build in monotonicity of your firing rate with a latent dimension. So you will have to introduce additional dimension to capture the non-monotonicity of the tuning curve. And the idea you will require, for example, with GPFA, as many dimensions as the number of kind of non-overlapping sets of tuning curves in this particular example. So kind of our answer to this, by modeling heterogeneity of neural tuning curves, we can reduce the dimensionality of their shared dynamics. Yes?
I have another question about cross-validation. It seems like you're doing 50-50 splits. Have you tried like a k-fold cross-validation with like 10 folds, which would give
Yeah.
parameters obviously there?
I don't have slide for this, but we tried it. And what, for example, the typical split is 80% for training, 10% for validation, 10% for testing. We did exactly that. So now your training folds become very correlated, right? So you keep shifting this 80% of the data along your data set. And the models which you will discover, I really regret I don't put a figure in there, because the model you discover basically the same overfitted model on all these multiple folds of validation. After that, you can average your models, but it doesn't really help.
If you have more and more data, you could actually fit more complex models in general. In your case, I think you were arguing that even if you have very little data in that regime, you could correctly find the true potential in this case. What's the relationship between the actual real model complexity and the amount of data you require?
Yeah, I don't have a plot for this, but you pointed to a good point. I talked a lot about overfitting. I didn't talk about underfitting. And if you have little data, you can underfit. And what happens with underfitting, you don't discover all the true features of your model, but then you start to overfit already. So we do see this behavior when we work with very small, like if we have only 1,000 spikes to fit the model, then we kind of and if our, as you say, potential is more complex, I only show double wells, but we can pick a more complex shape of the potential, then we do not discover all the potential features and we already start to overfit. So we kind of have some ideas of how to think about overfitting and have a criterion at least to detect that you underfitting. Because at this point, all of what I show will not tell you whether you underfit or not. So, but it's a separate project which is not ready yet. Yeah.
Just more quickly, a technical question. At some point, you had the curl flux portion, but later you only had the potential. Is your method restricted just to potential?
Yeah, so no, it's not restricted to potential. So in 1D, you only have potential, you have no curl. In reality, we fit forces. So potentially just more interpretable for visualization. It's kind of nice to look in wells than to look in the shape of forces. But we actually do fit forces, and I think when we do it in 2D, we will also kind of separate it in potential and curve flags because it's more interpretable.
Last question.
So you mentioned somewhere in the middle you talked about the rotational flow fields, but then that never came up again. Is that part of another feature?
Yeah, because I only focused on one-dimensional dynamics in these talks, so I didn't show anything about 2D.
Let's thank Tatiana one more time. And our next speaker up is Angela.